Hi, Andrew Wolf here. In this video I'm going to be talking about the physiology of hemostasis. Now I want to start out by just sort of talking about two different mechanisms of injury which both lead to different pathways in the formation of a clot. So first of all, we're going to talk about vessels that are just occurring on inside the vessel. So these are intrinsic to the vessel, and you may have heard of the term intrinsic pathway. Now I'm going to talk about the intrinsic pathway here, but I'm also going to talk about something that is separate from the intrinsic pathway called platelet plug formation. But these are both things that are happening inside an intact vessel. Okay, now on the other side, there are things that cause damage to tissues or disruption of the vessel, and this is going to stimulate the extrinsic pathway. So intrinsic, when I talk about things inside the vessel, I'm going to talk about platelet plug formation, and then I'm going to talk about the intrinsic pathway. And when I talk about extrinsic, I'm talking about things that that disrupt the vessel wall. Now, this is, interestingly enough, vessels are very, especially when you get down to the capillary level or the arterial or venule level, it doesn't take much of an injury to disrupt the vessel wall. So in reality, these two systems are almost always working concurrently. So we separate them in a way to understand them, but just remember that in real life it's rare for the intrinsic mechanisms to be initiated without the extrinsic mechanisms being initiated, and vice versa. Okay, so we're going to start out with injuries to the vessel wall. So we're going to take a small vessel here, and like a normal vessel, it is lined by the endothelium. So we have endothelial cells and a basement membrane. And remember, this is the lining of all blood vessels in the body have endothelial cells, and the heart itself have essentially the same cells. Now, endothelial cells, the endothelial lining, is the only surface known in the world that that blood can flow through without clotting. Now, we have been working for many years to create surfaces that that will not initiate clotting, but we haven't been able to do it yet. There is no system, there is no surface like endothelial cells. Endothelial cells are covered with sort of anticoagulant proteins that prevent the coagulation system from being initiated. And we know what some of these are, and I'm not going to get into it. We know what some of them are, and there are others that we don't understand yet. Now, inside the blood vessel, we've got little platelets, and if you remember our picture in the prior video on platelets, they're these little fragments of cells with sort of little spinous processes that stick out of them. Now, what activates platelets? Well, one of the main things that activates platelets is if we have a endothelial cell that is damaged and disrupted, this endothelial cell will secrete chemicals, will secrete a protein called von Willebrand factor. And von Willebrand factor activates platelets and makes them sticky. So this von Willebrand factor will be excreted by this injured cell and activate the platelets. So we have von Willebrand factor activates platelets, and the platelets form a plug. And the exact mechanism by which this occurs is proteins on the surface of platelets are actively binding with fibrinogen. and we get linkages between platelets. Now, fibrinogen is an important protein to know about. It is actually a inactive clotting protein in the body, and it will be activated later on to become fibrin. But in this instance, when it is binding platelets together and being part of the platelet plug, it is still in what is known as the fibrinogen or inactive form. So the only reason it's working here is because there are proteins here on the platelet surface that are now being expressed because of von Willebrand factor that cause the fibrinogen to bind with the platelets and create cross linkages, and that's how we end up with a platelet plug. And this is all occurring due to damage from endothelial cells inside the blood vessel. So again, this is platelet plug formation. Okay, now the next thing that can happen inside a blood vessel is initiation of the intrinsic pathway. Now this occurs, I'm going to draw a little blood vessel again, and typically with a nice healthy blood vessel, our endothelial cells make an impenetrable barrier. But what happens is, if we have significant injury, then the blood is exposed to this basement membrane below the endothelial cells. And in the basement membrane, particularly when it gets injured enough, we get these free ends of collagen proteins sticking into the lumen of the blood vessel. Now, collagen binds with protein in the bloodstream and causes them to change conformation. And what it does, the protein that it's binding with is called coagulation factor 12, otherwise known as Hageman factor. And Hageman factor becomes activated. It changes from factor 12 to factor 12A. And that just means that it has changed shape. So now it has an active binding site, and factor 12A binds with factor 11 and causes 11 to change to its active form, 11A, and then this cycle continues where 11A activates 9 to turn into its active form of 9A, and then 9A causes 10 to turn into its active form 10A, and then 10A causes prothrombin to turn into thrombin, and thrombin causes fibrinogen to turn into fibrin. And then fibrin creates long chains called a polymer, and then these long chains are sort of linked together in a fairly random fashion by a factor called 13A. I'll draw that in a different, and it creates cross linkages between the two, so the fibrin creates this mesh, this really complex sort of basket weave pattern, and that is what is the end of our clotting system. Okay, so now you don't need to know all the names of this, but I do want to make a couple points here. Number one is, why do we have so many steps here? We've got step 1 where collagen is binding with Hageman factor, stimulating the Hageman factor, and then Hageman factor is stimulating another factor and another factor and another factor and another factor. Now, actually, a lot of these active proteins are actually proteases, and what they do is they take a really complicated protein and they cleave it and break it up, and when it is cleaved, you end up with a active form with receptors on it that is now active. When it was put together, those receptors were just sort of bound within this huge matrix of a complex protein, but when it was cleaved by a protease, it was broken up so that one piece of it has active components to it. So a lot of these factors are actually proteases that are actually breaking up inert, inactive complex proteins into active parts. Okay, and if you can imagine, the reason why we have so many steps is because it's a system of amplification. Now, I used to use the example of a phone tree, but I think the technology is such that people aren't going to understand that as well. So I think the analogy that works well is a viral video, where you have, you've got Hageman factor that maybe creates a little video of some damage to a blood vessel, and it looks very fascinating. So Hageman factor goes out and tells 3 friends, and then those 3 friends tell 3 more friends, and those 3 friends tell 3 more friends, and as you continue, your multiplier gets bigger and bigger and bigger. So this is a system of, the reason why there are so many steps and the body has devised a system with so many steps is because each system amplifies the prior step by a significant factor. Now, interestingly enough, the intrinsic pathway that starts with collagen activating Hageman factor is very important to understand because it's involved significantly in the pathophysiology of thromboembolism. However, it's interesting to note that although this was actually the system that was first understood, but now we are realizing that it is the less important of the 2 major coagulation pathways. And we know that because if someone has a complete absence or a significant deficit of Hageman factor, they very rarely have bleeding diathesis, and they are able to survive a long and healthy life without Hageman factor. And that sort of underscores the fact that it is the extrinsic pathway that is the more important pathway in providing hemostasis. Okay, so let's talk about the extrinsic pathway. Okay, so the extrinsic pathway all starts out with a protein called tissue factor, and it's also known as factor 3. Now, this factor is a protein that actually can be that is found in 3 different places in the body. This is just a tissue cell, and the tissue factor is found inside the cells. It's found on some cell membranes, and it's also found in extracellular fluid. So there's sort of 3 different places where tissue factor is found. Now, so the tissue factor, again, like all the other coagulation proteins, exists in an inactive form. So what can activate it? Well, there's several different things. Inflammatory cytokines. This is important to realize because you actually can have inflammation without tissue injury, and this can cause initiation of the coagulation cascade. This is an important component to realize in sepsis, and it's what allows our body to create abscesses to sort of wall off infections, because you end up with, if you have a group of cells that are infected with bacteria, the fibrin will actually cause the area to be walled off over time, and you'll end up with an abscess. And the abscess is surrounded by a very strong wall that is made up of fibrin. Okay, so cytokines themselves can activate tissue factor and cause it to turn from its inactive form to its active form. Okay, the second thing that can activate tissue factor is cell injury. And the third thing is vessel injury or vessel disruption. And one of the major pathways that activates tissue factor here is just blood in the tissue. And I'm not sure exactly which protein in the blood is involved in this. There's probably several, but blood in the tissue will activate tissue factor as well. Okay, so let's talk about what happens. So any of these three things will cause tissue factor to turn into its active form. Now the active form factor 3A will cause factor 7 to turn into its active form, 7A, and then 7A will cause factor 10 to turn into 10A. Now you'll recognize this step here because 10 going to 10A also happens in the intrinsic pathway as well. So the rest of this pathway, 10 to 10A, prothrombin to thrombin, and fibrinogen to fibrin, is called the common pathway. Because this common pathway occurs in whether it's part of the intrinsic or the extrinsic pathway. Okay? So we're going to focus for now on the common pathway, and just keep in mind that once you get down to 10 being transformed to 10A, the rest is in common, and it's actually fairly simple. 10 to 10A, prothrombin to thrombin, fibrinogen to fibrin. Okay, so we're just going to kind of go through that again. Okay, so we've got 10A. 10A activates prothrombin, and prothrombin becomes thrombin. Now, thrombin is a protease, and what thrombin does, the way that it activates fibrinogen is, so fibrinogen is this long strand that has sort of a little globulin at the end, and then another fibrinogen here, long strand with a globulin at the end. And what thrombin does is it cleaves this fibrin. That means it breaks it into two, and what is exposed here is a receptor where fibrin can bind to itself. So you end up with this connection here where the fibrin binds to itself, and then you end up with these very long strands of fibrin. And if we so you end up with these long strands called fibrin polymers. And they're as strong as collagen strands, so they're very strong and straight proteins. And this doesn't finish the situation, so this is fibrin polymerization, but then we end up with lots and lots of fibrin polymers, and the final act in clot formation is activated factor 13 or 13A binds these together to create this thick weave of fibers. Okay, so that brings me to an end of this this brief video on the physiology of hemostasis and the coagulation cascade. Please let me know if you have any questions, and take a moment to provide feedback and a thumbs up or a thumbs down. And in our next video, I'm going to talk about the very important factors that control the coagulation cascade. I'll see you there.